Great.
I want all of you to picture the fireplace that is in front of us here and to be grabbing a cup of coffee or perhaps some warm cider as we settle in here for this discussion. I'm delighted to have this chance to speak with Professor Tsai and Professor Selko, two individuals who have certainly inspired work that I've done over the years, and I think as represented in the talks that they just gave, a tremendous technical and I think also conceptual tour de force. And so we'll ask a few questions here along the themes of past, present, and future, and thinking a little bit about the organizations that Dr. Selko and Dr. Tsai represent. And there'll be some time at the end here for some questions from the audience that I know you have burning as well from this. And so just to begin with here, when I think back myself about what I've learned about trying to develop therapeutics for challenging neurological and psychiatric disorders, I've realized that it's a team sport. We say this, but if I think about it, I think of the diversity of individuals that have to contribute from the early days of, for example, developing a CRISPR screen to testing molecules in vivo in animal models, and of course the future translational work that comes from that. And so I want to begin with just acknowledging both Professor Tsai and Selko have contributed to literally the training of hundreds of individuals across their time period. And collectively, I believe over 100,000 citations and over 500 different papers. That is a tremendous contribution to, I think, the field's knowledge and learning. And given those experiences, I want to maybe begin with Professor Selko here to comment on what you've seen about the dynamics in teams and where innovation and transformation really lies.
It's a very meaty question and hard one to answer in brief. I would say one of the things that worked well for us, both in the academic lab and in the biotech company that I helped to found and then have consulted to, is diversity. And I mean in all its forms. So obviously demographic, gender and ethnic diversity in terms of the folks that we attracted to the lab, and training backgrounds. One of the things that really made work go forward on the fundamental mechanism of Alzheimer's in our hands was the recruitment of electrophysiologists. So obviously a fundamental discipline for neurobiology, but one that many biochemically oriented labs, such as mine, were not expert on. And of course you can collaborate with others, but there's nothing like recruiting a bright electrophysiologist or a structural protein biologist or someone who does cryo-electron microscopy, some of these additional disciplines that most of us, at least not folks in my age, were trained in, but need to have in-house. And so in a biotech company, in a startup, and in academia, what really worked well for us was diversity of scientific backgrounds. And I can't tell you how important it was not to farm out some of our basic ideas and assays, but to have them working in-house, because when you're working with people directly, you troubleshoot every day, every hour of the day. So I think close connections with a series of different backgrounds in science, rather than just attracting people who are very similar to yourself.
Dr. Tsai?
Yeah, I really cannot agree more with Dennis. I really enjoy working with people from very diverse background, and nothing is more powerful to hear different voices and people come from different points of view, about a problem, and for me, I feel that especially I think fortunate is being able to recruit and work with not just neurobiologists and cell biologists and biochemists, but really people with engineering background. It just has been very, very initially refreshing, because the way they think is just so different from where, I received training. And they are very practical. They're there to get things to work, figure out, how to troubleshoot and how to, come out with a new device and new protocol, a new way of doing things. I want to share that it was, when I think back, very devastating during the pandemics. When the lab was shut down, and even though we could still meet via Zoom or other way of remote access mediated meeting, that innovation, in my opinion, for my lab, was gone for 2 years. It was very stark. But as soon as we were able to get back and see each other and discuss, all that new idea just pop out. So I really think interactions and interacting with people from diverse background is super important.
Great. So we're sitting here in the heart of Kendall Square, a place that some people would argue is the hub of innovation, and a place that many other organizations and cities dream of having such a concentration of activity. But when I remember Kendall Square, there was about 25 years ago only a movie theater here. And I remember very carefully at night coming here because there was simply nothing around here. And so, Dr. Selko, maybe you could just comment on how you've seen the landscape change through interactions with industry, thinking back to when you started to work on Alzheimer's disease versus what I imagine right now of just an inundation of opportunities.
Well, one of the things decades ago that was challenging was academic biotech, academic pharma interactions, because there was pretty much of a firewall between academics and companies. And I remember when I first started Athena Neurosciences, which was in the Bay Area, because that's where the investors were who wanted to do this work, we needed to have a special ombudsman appointed by Harvard and Mass General Brigham to check us out twice a year, come to the lab and ask my postdocs and graduate students, is Dr. Selko turning this into Athena East, basically, so you're doing the work for the company. And so I remember exactly who this fellow was. He was a neutral individual. He didn't have an axe to grind in terms of Alzheimer theories, and he did a very good job. And he said, no, it doesn't look like, there's plenty of basic science going on. There are theses that are being developed. It doesn't look like they're just trying to develop a biotech company. But one example of how things have changed is that now Kendall Square is full of biotech, and biotech, of course, is biology, and it's all of us. And so what I mentioned earlier about diversity, we've been able to attract people who have skills that they've developed in biotech companies, and they can collaborate with us, or they may come and work with us for a year or two. And that's very different than it was. We felt, like you did, going to the movie theater. I went to that theater too, and sometimes still do. One felt isolated and sort of empty and not integrated. And the academe was having trouble getting their neurons around how we would collaborate more intensively while still keeping intellectual honesty and financial honesty about what we did. So I think it's easier now to generate ideas. It's easier to put them into biotech and to get an exchange. And I think we had to make special efforts for that. I wanted to mention one other example, Steve, of getting people in your own group that have hands-on talent. Lipidomics, something that was beautifully illustrated by Louise's talk. And I think it's absolutely fascinating to see these oligodendrocytes that are APOE4 expressing or not, and how important it is to really drill down into the lipid analysis. But a great many of us would really not understand that at the molecular level. And so we, someone like Sarana Fanny, who came from Sue Lindquist's lab and joined us, and now has really gotten deep expertise, and a whole bunch of people working with her. So diversity is super important.
Dr. Tsai, maybe just to build on that, you've seen the concentration of companies and such around you, and but MIT made a decision to build the Picower Institute in ways in which it maybe anticipated the growth of neuroscience, or what led, I think, to the Picower and Department of Brain and Cognitive Sciences, really growing and thriving in this period.
Yeah, I think it has been very transformational. I remember, the Kendall Square area. I don't know how many of you were here 20 years ago. It was a dump, just very industrial. There are some candy factories, and so whenever you get close to this area, it just is very sweet smell. And there was nothing else. So yeah, maybe that theater was the exception. So I feel that our building, our neuroscience building, was probably one of the first wave of sort of new life science expansion that happened in the Kendall Square area. And I think the Broad Institute then opened after the neuroscience building, and then the Koch Institute, and then all the pharma companies all appeared. So in terms of the Picower Institute, we have grown from a handful of faculty members now to 16 strong. People doing very diverse different kinds of neuroscience, all the way from really studying the fundamental mechanism of learning and memory, all the way to looking at neuroimmune interactions and how the brain controls airways, and sort of very fascinating questions. One thing I think very kind of central to MIT, and I think the neuroscience community is kind of very closely following, is the revolution of artificial intelligence. So right now, right next to our building is the Schwarzman College of Computing. So I can see, our department is fully embracing machine learning and artificial intelligence, and I sort of feel that's going to be, the next big thing here in MIT neuroscience.
Really exciting. And maybe, Dr. Zucco, we'll turn back to the fact that you are part of a large academic medical center, as am I, at Mass General Hospital. How have you seen the sort of side of clinical care and training evolve in that same time period?
Very glad you asked that, because one of the things that builds strong teams is to have proximity between basic science labs. I mean, the techniques that I mentioned briefly today and that we use and Louis uses are fundamental to just basic biological science, but we apply them to disease models. And so it's been very good to actually be one of the few buildings, to my knowledge, in academic medical centers across the country that has clinics on the first 4 floors, and then has 10 floors of science. And we can literally take CSF samples from patients who are in our get our lumbar punctures on the first floor and bring them upstairs very easily. We can transport biopsies, and that is done all the time. There is a whole group under Howard Weiner's leadership that does microbiome research, and so we obviously need to get samples from patients all the time. So a lot more, I think, fluidity between the clinic and the basic science, which has to be good if it's well monitored and regulated for developing drugs, because rather than handing off our basic science to companies even as far away as Kendall Square for me, we're doing much more integration. And now it's been tremendously personally satisfying to beat my head against the amyloid wall for 4.5, 5 decades, turning out to be very bruised, but then going down to the first floor and actually writing an order for lecanemab and seeing how things work out, and that it's not as bad as actually even the side effect profile from the Clarity AD study suggested. So a lot of satisfaction being able to go back and forth between the clinic and the laboratory. And I think it works. And everything has to have guidelines and restraints. But if you follow those guidelines, the results are much more positive.
I imagine your use of rewarding is really an understatement. It must be thrilling, I think, to have that opportunity, to see that in the persistence of an idea and the persistence in the face of skeptics, probably on a daily, weekly, and monthly basis and such of it. And so just, amazing to see the persistence and power of that.
It's ironic that we actually, in academia, you may have this experience, we get hate mail. We get people who said, this is an outrage, you're completely misleading the world, blah, blah, blah. So not to go into it any further, but there is a sense of relief when you can look at objective data that thousands of people can say, yeah, it's really hard to explain that away, that it's an artifact.
We're in a safe space here, so.
Actually, I can second what Dennis just said. Not to compare our work, with Dennis, decades of contribution, but I don't know how many of you have heard. We recently came out with a non-invasive brain stimulation approach to reduce pathology and improve symptoms. And again, we actually have a small team of people working with human subjects, led by neurologists from Mass General. So it's really thrilling to be able to take the laboratory results and then apply it to humans immediately and see how humans respond, comparing animal data and human data. It's really extremely rewarding. I cannot stress more, and
So Dr. Simon, we could say a little bit more about that because you're anticipating my question about evolving modalities. And so in your talk, you had a few examples of some small molecules, and Dennis, you had a antisense or an RNAi agent against MAPT and some antibodies. But explain just, what made you think about a completely different direction of therapeutics that led to the breakthroughs that you have?
I think a lot of times what we do is really driven by data. You cannot outsmart, what have not happened. So, when I first came to MIT, I was very, happy to be able to interact with like a system neuroscientist. And then at the time, we took the brand new shining toy, which is optogenetics to manipulate neural activity. And so we just play with it. And we saw that when we drive a particular neural activity, we saw some very interesting results in the amyloid mouse model. We saw reduction of amyloid, in fact, very rapidly. So from there, we decided to test whether there can be a non-invasive way, safer than optogenetics to manipulate neural activity, and really from there. And now, really, we've seen some of the phenotypes, but what we really now want to understand is how does it work, right? What's the mechanism? How changing neural activity actually can lead to molecular and cellular changes in the brain. And like Dennis mentioned later on, we're also thinking about whether there are combination therapy that can further improve the efficacy. So, yeah.
How about you, Dennis, then, in terms of modalities? Then you've spent a lot of time thinking about antibodies. Are there others on the horizon that you are particularly excited about?
So I think anti-immunotherapy was a good example of something that wouldn't have been thought of traditionally as a way of curing or doing something for a brain disease. Antibodies wouldn't get in. By definition, blood-brain barrier keeps large molecules away. What turns out to be the case in that example is that They get in very inefficiently, but if you bathe the blood-brain barrier from the blood side chronically for months and years, enough gets in. Now we know there's a new method to do this, piggybacking on the transferrin receptor mechanism and using a so-called piggyback system that folks at Roche Genentech originally started talking about. And that looks like it's going to be a tremendous benefit over what I've said today, that you'll be much more efficient about that. But I think in terms of the broad question, Steve, about how do you start incorporating new technologies, I have to admit that it's difficult to break out of one's mold. So it's great to talk, as I have, about diversity and bringing in different perspectives. The very persistence that kept me focused on one goal, you know, from my perspective, has turned out okay so far. But I'm probably not going to be the best guy to invent the most powerful new novel therapy. And I think the Ways method, I actually contributed a patient to one of Cognito's trials because I thought this was a really cool way to use modern electrophysiology, essentially, to do something about it. And I have other patients I'd like to contribute. So it's a balance between sticking to your guns if you think that they're scientifically well-grounded and you look at the literature and you see what other smart people are saying, and then moving beyond that to something that's new. And so the thing that I really love is small molecule therapies that will supplant antibody treatment. Antibody treatments are very inefficient. It's great that we have something we can prescribe right now, but we're gonna, I think in 10 years from now or 15 years from now, either we'll have active vaccination where you make your own antibodies endogenously, if that can be done safely and controllably, or we will be returning to traditional pharmacology, which is small molecules. And we're working on that also, getting away from antibodies.
A bright future ahead. So, Dr. Tsai, you really beautifully showed the complexity of the human brain and the contributions of multiple cell types. And if I think back to the image, Dennis, you had of Alzheimer's neuropathology and the observations that can still be extracted out of that, do you think the future is going to lie within cell type specific therapies, or do you think there are still broadly acting targets?
Ooh, that's a very good question. I think that is actually one of the reasons why, you know, as I show you that we are doing genome-wide CRISPR screening and doing small molecule screens. We wanted to do all the different cell types because we precisely wanted to get at that question. We want to see, say, those genes upregulate and downregulate lipid accumulation in APOE4. Oligodendrocytes are the same pathways also regulating lipid disruption in astrocytes, in microglia, in blood vessel cells, or different pathways regulate lipid in APOE4 cells, right? So I think we need to at least get the basic understanding first to really figure out whether there will be some sort of global targets that we can go after to ameliorate APOE4 induced pathology and symptoms. So I think, my hope is that there are global targets. So, you don't have to combine 3 or 4 or 5 different therapies, which, I mean, there are problems, when you think about, interactions of, the different pathways and stuff like that. So I think it remains to be seen.
Maybe in the last few minutes here of asking some questions before we turn to the audience, I want both of you to project ahead into the future here, to think about when this same symposium is being held in 5 years or 10 years from now. What is it that you think is going to be the biggest breakthrough for treatment here? Will it be aspects of patient stratification, or you used the example, Dr. Selko, of the power of a series of positron emission tomography imaging agents to let you look at A-beta pathology while the patient was still alive? And so what do you think will really help us make a breakthrough in terms of the disease heterogeneity then?
I suspect it'll be a further application of the latest cutting-edge molecular biology to tweaking or interfering with specific gene products that almost certainly are driving disease. So rather than using antibodies, we may use antisense oligonucleotides, we may use CRISPR-Cas. I think it's amazing that, as I understand it, Ionis and Biogen have targeted the SOD1 form of ALS, and in those rare individuals who have that admittedly rare disease, they can get real clinical impact. So probably the future relies in precision medicine, to some extent individualized. The technique would be broad and global and used in many different cases, but targeting genes that you know cause the disease, at least for genetically based diseases. And I think that 5 and 10 years from now, we will have less Alzheimer's. And I think ultimately, you know, a lot of us are waiting to see whether treating pre-symptomatically, the way that hopefully everyone in this room is, even me, even I am still, not yet having so much disease pathology that you can't still talk a good game, I think we'll be able to nip the problem in the bud 5, 10 years before onset of symptoms, which means we will not see the emergence of clinical symptoms. People will have Alzheimer's, but it will be hidden. It'll be in their brains, and that's when we're going to treat it. And there's evidence already that that is working. I think when Risa Sperling's AHEAD study rolls out its data in 3.5 years, my guess is that while it won't be perfect, there'll be a bunch of people in that 1,200-person trial who will not have gone on to develop symptoms the way their mothers and fathers did.
Maybe,
In terms of the disease stratification, I sort of feel that, you know, in Alzheimer's, we don't have really stratification yet, although it's very prevalent in cancer. You know, different types of breast cancer take different kind of treatment. I often wonder whether if we would be in a position to stratify Alzheimer's disease, categorizing the patients into several different categories and developing more personalized medicine, whether that will be the way to go. For instance, you know, APOE4 carriers, whether they can constitute a sort of a category of Alzheimer's disease, and we know that they have much higher risk of developing Alzheimer's. So perhaps they will be the prime population that we can start a treatment earlier before the overt pathology develops.
Great. Maybe we can open the floor up here. If there are some burning questions in the audience, please come up to the mic here or from your seat.
Yeah. So I have a question, one for each. Have you looked at GLP-1 agonist in your mice? And what do you, I mean, you said at the beginning you treated 100, or you're treating 160 patients. That's a lot of data and wealth. One of the things that I'm curious about is adherence. Are people and their caregivers and their families supporting the treatments? Are they sticking around with it? Thank you.
Yeah, it's a great question. Semaglutide, so hot right now. It seems to be able to ameliorate anything, any symptoms, any disease. So, I mean, there is some, you know, evidence that disruption of metabolism, and some people consider, I guess, you know, Alzheimer's disease is some sort of type 3 diabetes or something along those lines. Definitely, there's a lot of evidence for disruption of lipid and glucose metabolism, disruption in different cell types. So I do think that semaglutide represents another class or another way of thinking about the disease. I think multiple groups are looking at semaglutide right now. I think it's exciting.
And I think in terms of translating the laboratory work to the clinic, it's been a challenge. There's been so much, our biggest problem is the lay press includes many folks who are not deep scientists writing articles. where they're off the mark. New York Times had a front page story that I've written a letter for. I don't know that it'll get published, where they made just gross mistakes. And so I got hold of the reporter and talked to him. He's a Pulitzer Prize winning investigative journalist who goes after tobacco companies and things of that sort. And I said, what about this? Well, he said, I think some people would disagree with you, Dr. Selko. I said, this is a basic fact. So one of the things has been convincing families and patients to not be as scared as some of the articles in the New York Times and elsewhere suggest they should be. And that's worked. And I mean, the people who come forward are not a perfect sample of the Alzheimer world out there. These are people who have the volunteer gene. They're willing to come in and try something. They're willing to listen to it. And we have very extensive discussions of risk-benefit ratio. So if you're really careful about talking with patients, they can become comfortable with it. And I had one fellow who was very skeptical and now is on his 20th infusion. He happens to have an E3, so he's at lower risk of ARIA, the major side effect we worry about, and he's doing very well. And now he wants to go to his house in Florida, and we've got to find him a neurologist down there that prescribes this also. So it's getting more widely accepted, but the uptake has been definitely slow because neurology traditionally, I can say this as a neurologist, is a therapeutically nihilistic field. The people who chose from medical school to go in that compared to surgery and cardiology and renal disease are the folks who said, I can figure out what's wrong in the brain, but I'm not going to worry about doing anything about it.
Please.
Hi, Christian Barescu from Visual Neuroscience. My first question, I'll have two to you, Li-Wei, is, as you and folks in the field think about deep profiling to, single cell data and finding subpopulations of potential therapeutics to map to subpopulations, and you're getting those from human brain, which is ideal. Have you found it challenging to also pair and get access to CSFs to try and see which of those markers can actually be detectable in an accessible tissue compartment for the drug hunters? Maybe think of an early biomarker that bridges the gap from, say, target engagement to the multi-month readouts that would be required, like modulation of amyloid. So that's my first question. If I get a chance for another one, I'll ask that.
Yeah, I think it's a great question. I think biomarkers, the more you think about disease, the more it appears that biomarkers almost everything. And I think the field now have now applied multi-omic analysis to biomarkers, but I think it's not enough. I think a lot more have to be done. I think the effort on proteomic analysis of the CSF samples and lipidomics, I think people started to do that. We recently just set up a collaboration with Oscar Hansen to access the Biofinder 2 cohort to look at lipidomics, especially the APOE4 carriers, really wanting to see whether there's anything we can identify as an early biomarker that we can then use to follow therapeutics. But I think CSF in general is kind of tough, right? Yeah. Because here, I think in the United States, people usually don't like to give their CSF samples. So in the end, you probably have to settle with plasma biomarkers, which apparently is a lot more complicated, obviously, especially when it comes to lipid. But I agree with you. It's something that really people, I hope that there will be a concerted effort from the government and private sectors to really look at the biomarkers seriously.
And if I may ask a forward-looking question to maybe both of you, but maybe directly to you, Dr. Selko, is, Dr. Tsai mentioned that, a lot of these genes that have low minor allele frequencies map to lipids, and many of them also map to the neuroimmune axis. And at the same time, these immunotherapies are all dependent upon effector function. And so they're engaging microglia. I wonder if you think that as these anti-amyloids that have FC effector function, will they alter the value proposition for those genetically implicated microglial targets, or does that inspire you that that cell population could be a good source for, as you pointed out, the important need for combination approaches to deepen the efficacy that we're seeing with the current therapeutics?
Yeah, I think it's the latter. I think there's a really good opportunity to modulate microglia without antibodies, so you won't care about effector function. There's already papers in the literature, and I'm proud of one of my closest colleagues and friends, Howard Weiner, who's a neuroimmunologist, MS expert, trained his attention to Alzheimer's and has used protoline, an adjuvant that is used in a number of vaccines, like Shigella vaccines, for example, for Army recruits, to show that it by itself, without an effector function, without an FAB region that targets amyloid, can clear amyloid, probably by activating peripheral monocytes that migrate into the brain and clear this. So I think there would be novel ways of modulating microglia that doesn't even rise to the level of, is there an effector function or not? We know that weak effector function, like crenezumab from Genentech, doesn't really do a lot in terms of clinical and amyloid removal. But I do think there will be non-immunoglobulin modulators, a lot of things. And Louis' lab has pioneered the idea of what microglial expression profiles indicate something that could be twisted into a positive or into a negative way. So the microglia are incredibly dimorphic in terms of their effect on Alzheimer pathology. I'll make one other comment about biomarkers, and that is we've been struggling to measure what I think could turn out to be a really useful biomarker, the oligomeric A beta that I touted earlier in mechanistic studies. We can measure tau, 12 varieties of tau, A beta 37 to 43, and we can't easily quantify these very hydrophobic dimers and oligomers that are really hard. And we've now got an assay that works very well in CSF, but we've got to get to plasma. So it doesn't work well in plasma, probably because the oligomers bind to a variety of proteins in the much protein-richer plasma. But in CSF, beautiful correlations between the level of oligomeric A beta, it doesn't see monomers, and tau and phospho-tau levels in those same CSFs. So if we could do everyone get an LP, we'd be there. But Louis is right, not everybody wants to get an LP.
So I think in the interest of time here, we're going to have to save your question, but Dr. Salko and Sai, I think we'll be around a little bit afterwards if there are further questions. And so with that, let me thank both of them for their amazing talks.